Very good evening to all the students of BIPC, especially to all the AP EAPCET aspirants of 2025. In this session, we are going to discuss about two chapters, that is photosynthesis in higher plants and respiration in plants, wherein from these two chapters definitely you are going to get 4 to 5 questions. Definitely you are going to get 4 to 5 questions. So stay tuned for another 40 to 45 minutes to get the complete concept of these chapters on your fingertips. So yes, let's get started. Firstly, discoveries of photosynthesis. Okay, discoveries of photosynthesis. I'll tell you a superb trick to remember all the discoveries. Mostly from the discoveries of photosynthesis, you will get match the following type of question or statement based question, wherein the discoveries are given at one column and the scientist names are given on the other column. So let's make this more clear just by getting the one standard formula for photosynthesis, that is 6 CO2 plus 12 H2O in the presence of sunlight and chlorophyll. It gives rise to glucose, that is C6H12O6 plus 6H2O plus 6O2, that is liberated as a byproduct. So this standard equation of photosynthesis is given by C.B. Van Hill. So from this standard equation of photosynthesis, we will recollect the complete discoveries just like this. So first, carbon dioxide is essential for photosynthesis is said by mole. It is known as mole's half-leaf experiment. Mole's half-leaf experiment. Okay, and water, sunlight are essential for the process of photosynthesis is said by Jan Ingenhousz. This is said by Jan Ingenhousz. By taking the model organism Hydrilla. Okay, and oxygen is evolved from photosynthesis. Okay, oxygen evolved from photosynthesis is also told by Jan Ingenhousz. Okay, and then if you see, glucose is formed at the end of photosynthesis. Glucose is the product of photosynthesis is said by von Sachs. Okay, so here oxygen is evolved from photosynthesis is said by Jan Ingenhousz, but oxygen is discovered by Joseph Priestley. Joseph Priestley in the year 1774. Okay, and oxygen is coming from water, not from carbon dioxide, is said by C.B. Van Hill. Oxygen is liberated, that is from water, is said by C.B. Van Hill. So yes, from this standard equation of photosynthesis, complete discoveries are on your fingertips. First, carbon dioxide is essential for photosynthesis is said by mole's half-leaf experiment, wherein by using KOH completely utilizes carbon dioxide, which at the end of the starch test will make the leaf half blue-green color and half pale color at the end of starch test. That experiment all of us know, just we have to remember who has said that carbon dioxide is essential for photosynthesis. Mole's half-leaf experiment by using KOH. And water and sunlight are essential for photosynthesis said by Jan Ingenhousz by taking the model organism Hydrilla. And then glucose is the product of photosynthesis said by von Sachs. About oxygen, it is discovered by Joseph Priestley by bell jar experiment, and oxygen is evolved from photosynthesis is said by Jan Ingenhousz, and it is coming from water, not from carbon dioxide, is said by C.B. Van Til. Okay, C.B. Van Til has conducted what photosynthesis beta? Bacterial photosynthesis. Bacterial photosynthesis, wherein he said that for bacterial photosynthesis, the electron donor will be H2S, whereby sulfur is being evolved. For oxygenic photosynthesis in higher plants, water is the electron donor, whereby at the end oxygen is evolved as a byproduct. These electron donors we have to remember. Bacterial photosynthesis, H2S is the electron donor. For oxygenic photosynthesis, H2O is the electron donor. This is the complete gist of your discoveries from photosynthesis in higher plants. Now, if we look into sunlight is essential for the process of photosynthesis is said by Jan Ingenhousz. But what spectra of sunlight is essential for photosynthesis, that is dealt by E.W. Engelmann. Okay, it is said by E.W. Engelmann. Okay, so this E.W. Engelmann, what did he do? He has took a filamentous algae that is Cladophora. Okay, filamentous algae that is Cladophora. A light which is exposed through prism is scattered as VIBGYOR, spectring out the 7 different colors. This spectra is exposed to filamentous algae called as Cladophora. Cladophora conducts oxygenic photosynthesis. Cladophora conducts oxygenic photosynthesis, wherein over this Cladophora aerobic bacteria are placed. Aerobic bacteria are placed. So here by placing aerobic bacteria over Cladophora, what happens is this spectra whichever is projected to this Cladophora, over which after some time you can see 2 peaks, wherein one peak which is showing the one peak which is in between 400 to 500 nanometers and other peak which is close to 700 nanometers. So 2 peaks are observed which is showing the maximum evolution of oxygen, maximum oxygen evolved from photosynthesis, their aerobic bacteria got accumulated. So once again if you see the light is projected through the prism, whereby it is scattered to VIBGYOR, which is further projected on the filamentous green algae that is Cladophora, which conducts oxygenic photosynthesis and liberates oxygen. By placing some aerobic bacteria over this Cladophora, you can observe 2 peaks like aerobic bacteria, this bacteria accumulation at 2 peaks. One peak is in between 400 to 500 nanometers and other at that is close to 700 nanometers. So with this experiment, T.W. Engelmann has drawn 2 spectra. One is absorption spectra, and the other is action spectra. Second is action spectra. So this absorption spectra will show you the different wavelengths of lights absorbed by different pigments. Action spectra will show you maximum wavelength of light absorbed by one pigment, okay, absorbed by the pigment, maximum wavelength of light absorbed or maximum photosynthesis conducted at that wavelength, that is shown through action spectra. Different pigments absorbing different wavelengths of light is projected in absorption spectra. So here based on the action spectra and absorption spectra, these are the 3 diagrams which are given, which are showing both action spectra and absorption spectra. So here absorption spectra, if you see in our academy book, three pigments are projected. One is chlorophyll A, second is chlorophyll B, and third is carotenoids. Okay, chlorophyll A, B and carotenoids. Just a second. Let's continue. Chlorophyll A, B and carotenoids. These are the three pigments that are shown in absorption spectra. So here if you see, chlorophyll A can show two peaks in between 400 to 500 nanometers. You can observe two peaks. Okay, chlorophyll A can show one peak and one more peak in between 600 nanometers to 700 nanometers. As such, you can observe two peaks of chlorophyll A. If you look into chlorophyll B, only one peak, one highest peak in between 400 to 500 nanometers and one small peak in between 600 to 700 nanometers. Carotenoids are absorbing light only between 400 to 500 nanometers. So here absorption spectra, if you can consider, the maximum wavelength of light is absorbed by chlorophyll A, where you can consider the two peaks at two different wavelengths. So here in the action spectra, if you see, only chlorophyll A peaks are shown. When you are projecting two action spectra and absorption spectra for rate of photosynthesis and absorption, two different graphs where one is showing the rate of photosynthesis, maximum rate of photosynthesis is shown in the action spectra. Okay, in action spectra, maximum photosynthesis rate is shown, that is by chlorophyll A, and absorption spectra of chlorophyll A, action spectra is coinciding with the absorption spectra of chlorophyll A. So with this graph, it is clear that three things you have to remember looking into this graph, that two peaks are shown by chlorophyll A and chlorophyll A is conducting maximum rate of photosynthesis. This is the second important point. And carotenoids are just in between, they absorb light in between 400 to 500 nanometers. And most important segment you have to remember is action spectra of chlorophyll A will coincide with the absorption spectra of chlorophyll A. Okay, absorption spectra of chlorophyll A coincides with the action spectra. That is the important point you have to remember from this. And who gave us action spectra and absorption spectra? That is E.W. Engelmann. Next, when it comes to chloroplast structure, okay, chloroplast structure, if you see, chloroplast structure, that is a double membrane bound organelle, where we will call chloroplast also as kitchen of the cell. Okay, chloroplast double membrane bound organelle, which is also called as kitchen of the cell, wherein it has two membranes and inside the fluid filled space is called as stroma. Sac-like structures inside the chloroplast are called as thylakoids. Okay, the sac-like structures are called as thylakoids. Sac-like structures called as thylakoids. Stalk of sac-like structure is called as grana. One grana is connected to the other grana by stroma lamellae. By stroma lamellae. One grana is connected to other grana by stroma lamellae. And chloroplast has a double stranded circular DNA. Chloroplast has a double stranded circular DNA. Double stranded circular DNA, which the ultimate goal of DNA we all know that is to synthesize proteins, right? So this double stranded circular DNA will synthesize the world's abundant protein, that is rubisco, here in the chloroplast. That is why chloroplast also got few synonyms like semi-autonomous cell organelle. This is also called as semi-autonomous cell organelle or self-replicating organelle or self-replicating cell organelle. Fine. So here from the chloroplast structure, what you have to remember beta, membranous sac-like structure is called as thylakoids, stalk of sac-like structure is called as grana, fluid filled space is stroma, and it has 70S ribosomes, 70S ribosomes, same like in prokaryotes, and it has double stranded circular DNA which is synthesizing the world's abundant protein inside the chloroplast, that is rubisco. So, these things you keep it in your mind with respect to chloroplast structure. Next, when it comes to the events in the photosynthesis, you have to remember photosynthesis is a sequential reaction which is in two ways, two reactions. One is light reaction or light dependent reactions, light reaction or light dependent reactions. Second type is dark reactions or light independent reactions. One is light dependent, other is light independent. So, from this light dependent reactions, if you consider, from light dependent reactions, 3 steps happen. You have to remember the site or the location of each event that is happening. So, the first is absorption of light in the form of photons, absorption of light in the form of photons. Second is photolysis of water, second is photolysis of water. Third is formation of assimilatory power, assimilatory power, formation of assimilatory power. Second sequence is light independent reactions wherein carbon dioxide is fixed or reduced to glucose. So, here what you have to remember is the sequence of reactions. Okay. First, absorption of light in the form of photons, it is done by thylakoid lamellae or thylakoid membrane. Okay. Thylakoid lamellae or thylakoid membrane, wherein thylakoid membrane has photosystems, thylakoid membrane has photosystems, that is PS1 and PS2. In this photosystems, you have light harvesting complexes wherein light is observed in the form of photons. So, this step where it is happening, thylakoid lamellae. So, this is happening in thylakoid membrane, or you will also call it as thylakoid lamellae. Second is photolysis of water. This takes place towards the inner side of thylakoid membrane towards the lumen. Okay, this takes place, photolysis of water takes place towards the inner side of thylakoid membrane towards thylakoid lumen. Next, assimilatory power. Formation of assimilatory power is nothing but formation of ATP and NADPH, formation of ATP and NADPH. The formation of ATP and NADPH is in stroma. This happens in stroma. Now, the carbon dioxide fixation to glucose, that is carbon dioxide reduced to glucose, this requires the assimilatory power which is formed at the end of light reaction. Okay, this reaction requires the assimilatory power which is formed at the end of light reaction. That is why we call this as light independent reactions, because it is not dependent on light but dependent on the products of light reaction. Calling light independent reaction as dark reaction is a misnomer. Okay. So, here carbon dioxide is reduced to glucose. This happens in stroma of chloroplast. This happens in stroma of chloroplast. Now, let us look into the light harvesting complex, which is the first step of light reaction. This happens in the thylakoid lamellae, wherein in light harvesting complex you have accessory pigments, accessory or antenna pigments, accessory or antenna pigments, which includes carotenoids and xanthophylls, chlorophyll B. This includes carotenoids, xanthophylls and chlorophyll B, where it absorbs the light at different wavelengths, and the ultimate acceptor or the primary acceptor will be chlorophyll A. So this chlorophyll A is called as reaction center, which absorbs maximum wavelength of light. So here this chlorophyll A will absorb maximum wavelength of light. Immediately after absorbing light, what happens is, inside the thylakoid lamellae, look into it clearly. So light harvesting complex, as you have observed in the previous slide, it has accessory or antenna pigments and a reaction center. Accessory or antenna pigments include chlorophyll B, carotenoids and xanthophylls, and reaction center has chlorophyll A. This is clear to you know. Now inside thylakoid membrane what happens is, so inside thylakoid membrane the photosystems are present like this. Here you have accessory or antenna pigments, antenna or accessory pigments, and reaction center, fine. Absorbing the light in the form of photons, photolysis of water takes place inside the thylakoid lumen. How the photolysis of water takes place? The water which is present inside the thylakoid lumen is photolyzed to 2 protons plus 2 electrons plus oxygen atom, okay, plus oxygen atom. This is how the photolysis of water takes place. The protons whichever are formed here will stay back inside the thylakoid lumen, but electrons will get excited to the reaction center. The electrons get excited to the reaction center. This is an uphill movement, and from here electron transport system starts. Okay. So what happens here is, from chlorophyll A, the primary electron acceptor is pheophytin. It accepts 2 electrons, okay, and then it gives to the next protein molecule, that is plastoquinone. From pheophytin the electrons are transported to plastoquinone. So this plastoquinone, immediately accepting 2 electrons, this takes protons from the stroma, that converts to plastohydroquinone. From this, H2 are transported into the lumen and 2 electrons are further given to cytochrome B6F complex. From that again it is lastly transferred to plastocyanin. This is photosystem 2. As such, you have one more photosystem, that is photosystem 1. Okay. So photosystem 2, maximum absorption of light is P680. Maximum absorption of light, maximum wavelength of light of reaction center is P680, and once the electrons are transported here, the primary electron acceptor is pheophytin, that gives to a proton carrier, that is PQH2, which again takes the protons from stroma and delivers to lumen. Next to cytochrome B6S, next to plastocyanin. Till here photosystem 2 will be done. So the electron movement will be in the downhill manner. Now again uphill you can see from plastocyanin to photosystem 1, okay, to photosystem 1, where at photosystem 1 the maximum wavelength is P700 nanometers. From plastocyanin the electrons are delivered to chlorophyll A, and then which has wavelength of P700, and then to ferredoxin, and then to ferrous sulfur complex. The ultimate or the last electron acceptor is NADP+. The ultimate or the last electron acceptor is NADP+, wherein accepting 2 electrons, this gets reduced to NADH plus H+, and this is by the enzyme NADP reductase. Fine. So with this is called as non-cyclic photophosphorylation, where this is in the uphill downhill manner. It is non-cyclic, and utilizing the sunlight, phosphorylation is happening. So this is called as non-cyclic photophosphorylation. From here, the main objective perspective you have to remember is photosystem 2 maximum wavelength of light is P680, photosystem 1 maximum wavelength is P700. That is one. Second type is, second objective type, that from photosystem 2 primary electron acceptor is pheophytin, last electron acceptor of photosystem 2 is plastocyanin, and primary electron acceptor ferredoxin for photosystem 1, and ultimate electron acceptor of this Z scheme is NADP plus. Okay, this is called as Z scheme. Fine. And here you have to remember what is uphill and downhill. And where is photolysis of water happening, beta? That is towards the inner side of the thylakoid membrane, inside the thylakoid lumen. This is what you have to remember. Now you can see NADH formation, but now we have to look into ATP formation. ATP formation. How ATP is formed by non-cyclic photophosphorylation. Here comes the concept of chemiosmotic hypothesis. Chemiosmotic hypothesis. Wherein in this chemiosmotic hypothesis, you have to remember 3 points. One is, first point that is that all the 3 points will be covered from non-cyclic photophosphorylation only. First point is what, beta? Due to photolysis of water, more protons are accumulated inside the thylakoid lumen, right? That is the first point. Second point is primary electron acceptor, that is pheophytin, is giving the electrons to a proton carrier, that is PQH2, where protons from the stroma are again taken and they are given to the lumen. Whichever least protons are present, they are utilized for making NADH by the enzyme NADP reductase. So by these 3 events, the proton concentration inside the thylakoid lumen will be increased and proton concentration in the stroma will be decreased. Yes or no? Because protons are taken by PQH2 and whichever little protons are there, they are added to NADH. With this, proton concentration inside the stroma will be reduced and inside the lumen will be increased. So this, from this concept of chemiosmotic hypothesis is derived. That is because of a proton gradient. That is because of what, ma? Proton gradient. Proton gradient in between stroma and lumen of the thylakoid. Proton gradient between stroma and lumen of the thylakoid. And chemiosmotic hypothesis requires a membrane, that is thylakoid membrane here. It requires a membrane, proton gradient and a pump, which is proton pump. Here it is called as ATP synthase. Okay, ATP synthase, that is F0, F1 pump. So here you can clearly look into proton gradient and a membrane is present, proton pump and ATP synthase. These are required for chemiosmotic hypothesis, wherein the protons from the thylakoid lumen are transported to, protons from the thylakoid lumen are transported to the stroma by F0, F1 pump, where F0 is inside the membrane and F1 is towards the stroma, which gives out 4 protons by a single pump, and inside the stroma you have ADP plus inorganic phosphate, which leads to the formation of ATP. This leads to the formation of what, beta? ATP. So this is what you have to remember with respect to chemiosmotic hypothesis, which is given by Peter Mitchell. And now comes the concept of dark reactions. Here quickly I will tell you what you have to remember. C3 cycle, C4 cycle and CAM cycle. C3 cycle occurs in the temperate plants, C4 cycle occurs in the tropical plants and CAM cycle in the desert plants. C3 cycle is also called as Kelvin cycle because it is discovered by Melvin Kelvin. what is the primary carbon dioxide acceptor in C3 plants? That is, primary carbon dioxide acceptor is RUBP, and it is catalyzed by world's abundant enzyme, that is Rubisco. Whereas in C4 plants it is also called as Hutchins-Slack pathway, and here the primary carbon dioxide acceptor is phosphoenol pyruvate catalyzed by peptidase enzyme. And CAM cycle and C4 cycle are one and the same, but in CAM cycle only mesophyll cells are involved, but in C4 cycle both mesophyll cells and bundle sheath cells are involved. So whatever points are I am saying, beta, keep it in your mind more strongly. And in C3 cycle the primary organic intermediate that is formed is phosphoglyceric acid. C4 cycle primary or the first stable organic intermediate is oxaloacetic acid. Here also the first stable organic intermediate is oxaloacetic acid. And here if you see in C3 cycle we have discussed the primary carbon dioxide acceptor enzyme, where it forms two molecules of phosphoglyceric acid, and these two molecules of phosphoglyceric acid are reduced. They are reduced to two molecules of glyceraldehyde 3-phosphate. This I am talking about 1 carbon dioxide fixation. For 1 carbon dioxide fixation, this reduction step, for 1 carbon dioxide fixation, reduction from 2 molecules of phosphoglyceric acid to 2 molecules of glyceraldehyde 3-phosphate requires 2 ATP and 2 NADPH. 2 NADH. This you have to remember. And if it is for 12 phosphoglyceric acid molecules, that is 12 ATP and 12 NADPH. Okay. Now if you look into the sequence of this regeneration, in regeneration you have 7 steps. From this the most possible objective are like which is the highest carbon organic intermediate, that is pseudoheptulose. 5 carbon compounds if you see, erythrose and 4 carbon compounds that you can see, cellulose and erythrose, like that. They will ask you the questions with regards to organic intermediates. For 1 carbon dioxide, for regeneration, 1 ATP is required. For 6 carbon dioxides or for the formation of 1 glucose, 6 cycles are required, that is 6 carbon dioxides are required. Okay, how many? 6 carbon dioxides are required, 18 ATP and 12 NADPH. Keep that in your mind, beta. How many carbon dioxide fixations have to be done for 1 glucose molecule formation? 6 carbon dioxide fixations, 6 cycles. How much ATP? 18 ATP and 12 NADPH. Keep that in your mind. Next, coming to C4 cycle. In C4 cycle, if you see, first you have to analyze the Kranz anatomy of bundle sheath cells. Kranz anatomy of bundle sheath cells. What is it? You know, these are mesophyll cells, and around the vascular bundles, few cells are surrounded along the vascular bundle in the form of a wreath, or garland. There is no intercellular space between these cells, and these cells are called as bundle sheath cells. There is no intercellular space between these cells. These cells are called as what, beta? Bundle sheath cells. So this bundle sheath cells have chloroplast, but there is no grana in the chloroplast. That is why you will call it as agranal chloroplast. Okay. In this agranal chloroplast, C3 cycle takes place, and in the mesophyll cells, C4 cycle takes place. Mesophyll cell has granule chloroplast. Now let us see how the process is activated. First, carbon dioxide fixation takes place in the mesophyll cells by the primary acceptor, that is PEP, and the first organic intermediate formed is 4 carbon compound, that is oxaloacetic acid, which forms malic acid or ascorbic acid. This is passed on to the bundle sheath cells. Inside bundle sheath cells, if you see, this 4 carbon compound is decarboxylated to 3 carbon compound pyruvate and 1 carbon dioxide. This 1 carbon dioxide will activate Kelvin cycle, and this 3 carbon pyruvate will regenerate the PEP back to the mesophyll cells. And how does the malic acid or ascorbic acid is passing on to the bundle sheath cells as through plasmodesmatal connections. This is through plasmodesmatal connections in between the mesophyll cells and bundle sheath cells. This is through plasmodesmatal connections between mesophyll cells and bundle sheath cells. So now if you see, for one carbon dioxide fixation in C4 cycle, it requires 5 ATP because 2 ATP for C4 cycle and 3 in C3 cycle. On total it is 5 ATP and 2 NADPH. For one glucose formation, it is 6 carbon dioxides or 6 cycles are required, which needs 30 ATP and 12 NADPH. So this energetics is very important both with respect to C3, C4 and CAM cycle. Okay. So now with respect to CAM cycle, the important thing we have to remember is photoactive stomata. In normal plants you have photoactive stomata where stomata opens during daytime, but in CAM plants it is photoactive stomata where stomata opens during night time and closes during daytime. So here carboxylation or carbon dioxide fixation happens during night time and the 4 carbon compound, that is oxaloacetic acid, is turned to malic acid. It is stored inside the vacuole and during daytime decarboxylation occurs and this runs the Kelvin cycle. Carbon dioxide will run the Kelvin cycle. So here the energetics for C4 cycle and CAM cycle is one and the same, but here everything happens inside the mesophyll cells only. Carboxylation, first carbon dioxide fixation happens during night time. That is what you have to remember with respect to CAM cycle. Now there is an important concept, that is photorespiration, which is also called as C2 cycle beta. So this C2 cycle is operated only in the C3 plants. C2 cycle is operated only in the C3 plants, wherein inside C3 plants, that is temperate plants, what happens is RUBP, okay, Rubisco is the enzyme, we all learned, right? Rubisco is the enzyme. This has equal affinity of binding for carbon dioxide and oxygen. Actually more affinity for carbon dioxide and little less for oxygen. But whenever oxygen concentration is more in the intracellular level, what happens is carbon dioxide will be accepted by primary carbon dioxide acceptor, that is RUBP. This is Rubisco whenever it is binding with oxygen. Whenever carbon dioxide is not there, only oxygen is present. At that time what happens is one molecule of phosphoglyceric acid, which is 3 carbon, and one molecule of phosphoglycolate, that is 2 carbon, is formed. One molecule of phosphoglyceric acid and one molecule of phosphoglycolate is formed beta. So here if you see, with one molecule of phosphoglyceric acid, it will not happen, C3 cycle will not be continued. So to generate one more molecule of phosphoglycerate, this has to, phosphoglycolate has to travel through 3 organelles. It has to travel through 3 organelles, that is chloroplast, peroxisomes and mitochondria. So this is very important. How many organelles are involved in photorespiration? 3 organelles: chloroplast, peroxisomes and mitochondria. So here if you see, in the complete process of photorespiration, there is no amount of sugar is synthesized, no amount of ATP is produced, rather carbon dioxide is released. So that is why this process is said to be a wasteful process, which is operated only in the C3 plants. It is not operated in C4 plants or CAM plants. Do remember, from this definitely you will get a statement based questions. Most highly we can expect from this type of questions from photorespiration. Why is it called as wasteful process? Because no sugar, no ATP synthesis, rather carbon dioxide is released out through the process of photorespiration. How many organelles are involved and what are they? Chloroplast, peroxisomes and mitochondria. This is the gist of photorespiration beta. I have to run fast because we have to cover the respiration chapter also. So this is a fast track series, just grasp as many points as you can. Factors affecting photosynthesis, as we all know, external factors and internal factors both affect the process of photosynthesis. External factors include light, temperature, water, and internal factors include age and orientation of leaves, chloroplast synthesis and chloroplast number. When there are multiple factors involved in one biochemical reaction, here comes the concept of Blackman's law of limiting factor. Okay, that is Blackman's law of limiting factor. Blackman's law of limiting factor. So here in this, what does this law say? Blackman's law of limiting factor. It says that when any biochemical reaction is carried through multiple factors, the process is determined by the factor which is nearest to the low value, okay, which is nearest to the minimal value, that decides the whole process of biochemical reaction. That is Blackman's law of limiting factor, do remember. So now if we look into the first external factor, that is light. Okay, first external factor, what is it, ma? Sunlight. First, that is sunlight. So you have to remember this point, that is 10% of total sunlight will lead to saturation. Just 10% of total sunlight is enough for the process of photosynthesis. So initially, as the light intensity is increasing, photosynthesis rate also increases. So here on the x-axis you can see, light intensity, on the y-axis rate of photosynthesis. Initially, as the light intensity increases, rate of photosynthesis also increases, but it reaches to the saturation point at 10% of the total sunlight. After that, even if you increase the light intensity, there will not be any increase in the rate of photosynthesis. That's it, this you have to remember. Next, when it comes to temperature, okay, temperature, the optimum temperature for C3 plants, is 25 to 30 degree Celsius. Optimum temperature for C4 plants, which are tropical plants, that is from 30 to 40 degree Celsius, or you can say precisely 30 to 35 degree Celsius is the optimum temperature. Here, when temperature comes to the picture, the enzymes like Rubisco and peptidase, these enzymes are temperature sensitive. So temperature affects dark reactions. And lastly, carbon dioxide, okay, carbon dioxide. So carbon dioxide, the optimum level is 0.03%. As you increase it to 0.05% and above 0.05%, with increase in the rate of photosynthesis at low light intensities, even if you give high carbon dioxide concentrations, plant will not respond. Carbon dioxide concentration will be affected only at high light intensities, okay, up to 0.05%. So here you have to remember, for C3 plants, the carbon dioxide saturation point is up to 450 microliters per liter, okay, and for C4 plants the maximum carbon dioxide saturation point is 360 ml per liter. Okay, this you have to remember, the saturation points both for C3 and C4 plants. Now, after completion of photosynthesis, we are looking into respiration. Here each and every point that I am saying, fix it in your mind beta, all that we will discuss in complete fast series. Firstly, how do plants breathe? Through two structures, that is lenticels of stem and stomata of the leaf. Through these structures the plants breathe, or they will exchange the gases. How do they respire? Is there any strong respiratory system as how you will see in the human beings? No, the gaseous exchange of all cells is very minimal. There is no, every plant part will take care of its own gaseous need, and there is no complex gaseous exchange mechanism as how there is in human beings. Here you have to just remember what are the breathing breathing structures of plant, that is stomata of leaf and lenticels of stem. Now here comes the concept of cellular respiration, and the difference between breathing and respiration. Breathing is nothing but exchange of gases. Respiration is oxidation of macromolecules like proteins, carbohydrates and fats. What is oxidation? Breakdown of the complex food material, that comes under respiration. So, cellular respiration, oxidation of macromolecules inside the cell and releasing out the cellular energy, that is ATP. Releasing out the cellular energy, that is ATP. This is called as cellular respiration, which happens inside the cell. So, what is the cellular energy? That is ATP. And the substrates whichever are involved in the respiration, like carbohydrates, proteins, fats, these are called as respiratory substrates. They are called as respiratory substrates. Okay. So, are you clear with the terms of respiratory substrate, cellular respiration and cellular energy? Now, what happens inside the cell of both prokaryotic cell and eukaryotic cell? Let us discuss. So, what happens in a prokaryotic cell? You can see it over here. Prokaryotic cell and eukaryotic cell, what happens? Or else you can clearly look into two types of respiration. One is anaerobic and the other is aerobic. So, here glucose, firstly, inside every cell, glucose is partially oxidized or incompletely oxidized to 2 molecules of pyruvate. This is the process called as glycolysis, happens in the cytoplasm of the cell. Okay, this happens in the cytoplasm of the cell. So, this pyruvate, if it is conducting anaerobic respiration, that is in the absence of oxygen, when oxygen is absent, this pyruvate can conduct 2 pathways. One is lactic acid fermentation and the second is alcoholic fermentation. These are the 2 possibilities of anaerobic respiration. In aerobic respiration, the pyruvate molecule will directly enter into mitochondrial matrix and it will undergo a link reaction or oxidative decarboxylation, forming oxaloacetic acid, thereby Krebs cycle, that is TCA cycle, and finally in the inner mitochondrial membrane, what happens? Electron transport system will be activated. So, here in the anaerobic respiration, that is in the absence of oxygen, 2 possibilities are there. One is lactic acid fermentation, second is alcoholic fermentation. In the presence of oxygen, pyruvate molecule directly enters into the mitochondrial matrix and first oxidative decarboxylation TCA happens inside the mitochondrial matrix. Electron transport system occurs in the inner mitochondrial membrane. Now, if we look into the glycolysis process, okay, glycolysis process. Glycolysis is also called as EMP pathway, which as it is given after the scientists Emden, Meyerhoff and Parnas. So, here first if you see first step, glucose to glucose 6-phosphate by hexokinase. Glucose 6-phosphate to fructose 6-phosphate by hexose phosphate phosphohexokinase. This is by phosphohexokinase. So, here most importantly you have to remember the enzymes of each step and the energetics involved. If you clearly see at first and third step, 2 ATP molecules are utilized. It utilizes 2 ATP molecules. At 6th step, that is glyceraldehyde 3-phosphate to 1,3-bisphosphoglyceric acid, 1,3-bisphosphoglyceric acid, 2 NADH. If we consider from one glucose molecule, 2 NADH are released. 2 NADH are released. And at 7th step and 10th step, okay, this is substrate level phosphorylation, wherein on total for one glucose molecule 4 ATP are being released into the cytoplasm. Of this 4 ATP which is released, 2 ATP are utilized initially at the preparatory phase. The first 5 steps are called as preparatory phase. The next 5 steps we are calling it as payoff phase. So, in the first 5 steps, that is preparatory phase 1 and 3, 2 ATP molecules are utilized from the cytoplasm, and at 6th step 2 NADH are being released into the cytoplasm. At 7th and 10th step 4 ATP molecules are released. Of this, 2 ATP are being utilized by the preparatory phase. So, here for the glycolysis, net ATP will be 2 ATP, net ATP will be 2 ATP molecules, and the total ATP released, that is for 2 NADH, 2 into 3, we will call it 1 NADH is equal to 3 ATP. So, 2 NADH, it is 6 ATP plus 2 net ATP. So, total ATP is 8 ATP that is released through glycolysis, and for each and every step, that is very important to remember the names of the enzymes, and which is the rate limiting step here, that is the third step, and the enzyme that is catalyzing is phosphofructokinase. All the 10 steps should be in your mind beta, including the enzymes, and most importantly the energetics, net ATP, total ATP that is released, and at which step what sort of energy is being released. Now, these 2 molecules of pyruvic acid, if they are destined for anaerobic respiration, 2 types are possible, I said, you know, alcoholic fermentation and lactic acid fermentation. If it is alcoholic fermentation, first pyruvate is converted to 2 molecules of acetaldehyde by pyruvic acid decarboxylase, and acetaldehyde to ethanol by alcoholic dehydrogenase, So, here at the end ethanol and carbon dioxide are released. Here it requires 2 NADH. This 2 NADH which is utilized at this step is taken from glycolysis, where NADH is released at the 6th step of glycolysis, that is utilized by alcoholic fermentation, and this releases 2 NAD plus, that is utilized by glycolysis. So, here there is no amount of energy that is released through the process of fermentation. There is no energy released through the process of fermentation. Okay. So, what is the amount of energy that is released due to anaerobic respiration is just 2 ATP. That 2 ATP is also from the glycolysis, that is the net ATP of glycolysis. From fermentation, there is no amount of energy that is released. Okay. But here maximum alcoholic accumulation above 13% will cause the death of yeast, that you have to remember, will poison the cells. Alcohol percentage, if it is above 13%, that will poison the cells. And from one glucose molecule, the energy that is released due to alcoholic fermentation or lactic acid fermentation, that is through anaerobic respiration, the amount of energy released from one glucose is less than 7%. It is less than 7%. These 2 points you have to remember, very much important. Next, if you see lactic acid fermentation. So, here glucose molecule is converted to 2 molecules of pyruvate, that is in glycolysis, and the same way that NADH, okay, NADH is being synthesized, which is synthesized from the 6th step of glycolysis, is utilized in lactic acid fermentation. Here NAD plus is utilized for glycolysis again. So, here also net ATP released is just 2 ATP from the process of lactic acid fermentation. Lactic acid fermentation takes place in prokaryotic cells like lactobacillus. In eukaryotic cells, especially in the human beings, it occurs in the muscle cells. Muscle cells. Whenever we are doing vigorous exercise or workout, at that point of time lactic acid fermentation is activated in the muscle cells, wherein due to the accumulation of lactic acid in the muscle cells we get muscle cramps after the exercise. That is the major reason, remember that. And the same point is applicable, less than 7% of energy is released from the glucose through lactic acid fermentation also. So, and next if you see aerobic respiration. If at all in aerobic respiration, what happens is, if you consider this as a cell, through the process of glycolysis, glucose is converted to 2 molecules of pyruvic acid. Glucose is converted to 2 molecules of pyruvic acid. This is called as incomplete oxidation or partial oxidation. This pyruvic acid will directly enter into the mitochondrial matrix. This will directly enter into mitochondrial matrix. So, if you consider mitochondria, it has 4 parts. One is outer mitochondrial membrane, second is inner mitochondrial membrane, in between those two you have intermembrane space, and at the center mitochondrial matrix. Pyruvic acid will directly land into mitochondrial matrix. Inside mitochondrial matrix, if we consider 1 molecule of pyruvic acid, that is for one cycle, along with coenzyme A and NAD plus, okay, in the presence of pyruvic acid dehydrogenase. Pyruvic acid dehydrogenase, it will synthesize 1 molecule of acetyl-CoA and NADH plus H plus. Okay, this is being synthesized inside the mitochondrial matrix. This acetyl coenzyme A will start activating tricarboxylic acid cycle inside the mitochondrial matrix. So, inside mitochondrial matrix what is happening beta? Pyruvic acid is landing inside the mitochondrial matrix, and inside the mitochondrial matrix what is happening? The tricarboxylic acid cycle is getting started after oxidative decarboxylation. Here in TCA cycle, citrate first formed is citrate by oxaloacetic acid and acetyl-CoA. The very first organic intermediate formed is citrate. Then it is converted to isocitrate by aconitase enzyme. Isocitrate through the enzyme isocitrate dehydrogenase, and it will release NADH here. So, I think all of you might be aware of TCA cycle. Here I will clearly look into those objects wherein energy is being released beta. So, here if you see isocitrate is isocitrate to alpha ketoglutarate. For 1 molecule of glucose if you consider, 2 NADH are released, and from alpha ketoglutarate to succinyl coenzyme A, 2 NADH are released, and other point, that is last step, from malate to OAA, 2 NADH are released. At one point FADH2 is released, that is succinate to fumarate, and at one place substrate level phosphorylation happens, that is from succinyl CoA to succinate, where ATP is being released. Okay, this happens in mitochondrial matrix. So, if we have to calculate the energetics, we will if we have to calculate the energetics of aerobic respiration, first is glycolysis, where the net ATP is 2 ATP and 2 NADH are released at 6th step, which into 3, that is 6 ATP on total. Okay, but this NADH to be transported into the mitochondrial matrix, for 1 NADH 1 ATP molecule is utilized by Here one ATP molecule is utilized by glyceraldehyde 3-phosphate shuttle system, glyceraldehyde 3-phosphate shuttle system. One molecule glyceraldehyde 3-phosphate shuttle system, the energy is being delivered, okay. That is for one NADH molecule it will utilize 1 ATP. So, on total we have to calculate, we have to eliminate 2 ATP here as they are being utilized by G3P shuttle system, 4 ATP on total. I hope you have understood. From glycolysis 2 NADH are released, but to the NADH to be carried to the mitochondrial matrix, one shuttle system is activated, we will call it as glyceraldehyde 3-phosphate shuttle system. It will utilize 1 ATP to transfer 1 NADH. So, to transfer 2 NADH, this shuttle system is utilizing 2 ATP, okay. Shuttle system is utilizing 2 ATP, so that it will pass on just 4 ATP. Second is oxidative decarboxylation. Oxidative decarboxylation for one glucose molecule, 2 NADH are being synthesized. 2 NADH means 2 into 3, that is again 6 ATP. And lastly, if we take TCA cycle, that is tricarboxylic acid cycle, you can see at 3 points, that is 6 NADH into 3, that is 18 ATP for one glucose molecule, that is 2 cycles of TCA, and 2 FADH2, that is 2 into 1 FADH2 is equal to 2 ATP. So, 2 FADH2, that is 4 ATP, okay, and 2 ATP molecules, that is from succinic acid to succinyl-CoA to succinic acid, succinyl-CoA to succinic acid. So, now if we count all this, we will get 36 ATP. So, in your exam if there is a question of how much energy is being released from aerobic respiration in a eukaryotic cell, if that is your question, you have to go for 36 ATP. If they are generally asking you how much ATP is released from aerobic respiration, then you can go for 38 ATP, because in eukaryotic cells G3P system shuttle system is activated, you have to eliminate 2 ATP. Now, we have to look into ETS, electron transport system, which is very much important. In here in the NEET examination, the complexes are asked. In TS EAPCET also there was many questions on complexes, at least 2 to 3 questions I have found out in different papers. So, here if you see, we have 4 complexes, that is electron transport system, which is activated in the inner mitochondrial membrane. So, here in the inner mitochondrial membrane you have 4 complexes beta, that is complex 1, 2, 3 and 4. Complex 1 is NADH dehydrogenase. Inside mitochondrial matrix there are so many protons present. So, NADH is dehydrogenated to NAD++ H+. 4 protons are pumped to intermembrane space and 2 electrons to the molecules flavin mononucleotide, that is FMN and FES. 2 electrons are passed from that to ubiquinone. And next again plus are being carried from complex 2 by succinate dehydrogenase, that is FADH2 is dehydrogenated to FAD+. So, here FAD+ to ferrous sulfur complex, here both electrons and protons are given to the mobile electron carrier, that is ubiquinone. Here only electrons are given to ubiquinone. From here both electrons and protons are given to third complex, that is cytochrome Bc complex. From here you can see cytochrome B, ferrous sulfur complex and cytochrome C. Again here you can come across a mobile electron carrier, that is cytochrome C, which is on the outer side of the inner mitochondrial membrane. This gives the electrons to complex 4. Here you can see copper molecules and cytochrome A, A3. ultimate electron acceptor is oxygen. The ultimate electron acceptor is oxygen molecule. That is the ultimate electron acceptor, which leads to the formation of water here. So, here what you have to remember from ETS beta, 4 complexes: complex 1 NADH dehydrogenase, complex 2 succinate dehydrogenase, complex 3 cytochrome BC complex. Here what is the mobile electron carrier? Ubiquinone, and in all these 3 complexes ferrous sulfur complex is present. And from complex 3 to 4 there is a mobile electron carrier, that is cytochrome C. From cytochrome C it is given to copper ions, and the ultimate electron acceptor is oxygen molecules, which leads to the formation of water. After this complete ETS process, there is proton gradient generated between intermembrane space and matrix. So, in intermembrane space you can see the accumulation of protons, that is 4, 4 and 2, like accumulation of protons due to ETS, and less protons in the matrix. Pump which is generated, that is ATP synthase, F0 F1 pump, that will lead to the formation of ATP inside the mitochondrial matrix. This is a very important process, you have to remember beta. Few more objective like about different complexes, mobile electron carriers, ultimate electron acceptor, and ATP is formed inside mitochondrial matrix. And now if you look into amphibolic pathway, respiration is said to be amphibolic pathway because amphibolic pathway because it involves both anabolism and catabolism. Catabolism, respiration is the catabolism, that we all know. How is it involved in anabolism? The respiratory the organic intermediates at different levels, like for example alpha ketoglutaric acid, succinyl coenzyme A, oxaloacetic acid. These are the different organic intermediates that are formed at different levels of TCA cycle or oxidative decarboxylation. This organic intermediates forms the starting step of biosynthesis of some secondary metabolites or some accessory pigments like carotenoids. So, that is why this is also called as amphibolic pathway, because the organic intermediates at different levels of this aerobic respiration, they are involved in the biosynthetic pathways of secondary metabolites or some pigments like carotenoids. Now, if you look into different respiratory substrates, how they are joining the cycle. To all the respiratory substrates like fats, carbohydrates and proteins, the common pathway is Krebs cycle, that is TCA cycle is common for all the respiratory substrates. Most important beta, you have to remember, for all the respiratory substrates the common pathway is TCA cycle. How fats are joining? Fats breaking down to fatty acids and glycerol. Fatty acid links up to TCA cycle by acetyl-CoA and glycerol through DHAP, that is the beginning of payoff phase in the glycolysis. Next, carbohydrates through glucose 6-phosphate, fructose 6-phosphate, this we all know, that is glycolysis, oxidative decarboxylation and TCA cycle, this we all know. Next, proteins breaking to amino acid, and that leads to pyruvic acid. Pyruvic acid will join the cycle of aerobic respiration. This is how different respiratory substrates are joining the process of aerobic respiration. Last segment of respiration in plants, that is respiratory quotient beta. Respiratory quotient, where volume of carbon dioxide evolved by oxygen consumed. Respiratory quotient is what? Volume of carbon dioxide evolved by oxygen consumed. So, here if you clearly look into, if glucose is the respiratory substrate, 6 carbon dioxides evolved and 6 O2 are utilized. So, RQ value will be 1. If fats, for example tripalmitin, for fats, any of the fat, it will be RQ value will be less than 1, okay, it will be less than 1 only, RQ value will be less than 1 for fats. For proteins, RQ value will be around 0.9. For organic acids, if you take organic acids, the RQ value will be greater than 1 for around 1.3 to 1.4. Okay. And last, if you consider for anaerobic respiration, the RQ value will be infinity. For anaerobic respiration, the RQ value will be infinity. Why is that so? Because 2 carbon dioxides are evolved through alcoholic fermentation and no oxygen is utilized. Something by 0 is infinity. So, these are the RQ values you have to remember. For glucose it is 1, for proteins it is 0.9, for fats it is 0.7, organic acids it is 1.3 to 1.4, anaerobic respiration it is infinity. So, with this we have completed the fast track revision of both photosynthesis and respiration. Stay tuned for more important concepts revision for especially AP EAPCET 2025. Thank you.